Hello, my name is Jens Schwambon. I'm the CEO and co-founder of OrganoTherapeutics. We use human Parkinson's disease patient-specific midbrain organoid technology to develop new therapeutics against Parkinson's disease. The human population on this planet is aging. You can see here an age distribution in the 1960s in comparison to the 2060s. And what you can very clearly see is that the amount of elderly people is dramatically increasing. This is not only a problem of the so-called Western world, but a truly global problem, as you can see here in the graphics on the right side. Together with aging, age-associated diseases become a more and more prevalent problem. And among those age-associated diseases, we are focusing on Parkinson's disease. Parkinson's disease is a neurodegenerative disorder where a particular type of nerve cells in the brain degenerates. These nerve cells, dopaminergic neurons, shown here in red, control, among other things, motor behavior. And therefore, upon their degeneration, one of the clearest symptoms is shaking, tremor, rigidity. I'm sure you have all seen those symptoms in Parkinson's patients before. Currently, we only have symptomatic treatments against the disease. We have nothing that prevents those dopaminergic neurons from dying. And the reason for that is that we are over-relying on the wrong model systems. So we do, in the last decades, a lot of research on mice, but mice are clearly not human. If you just look at the brains down here, there's millions and millions of years of evolution between the mouse and the human brain. The human brain is nearly 4,000 times bigger and way more complex than the mouse brain. Hence, using these systems so far has not led to a real therapeutic against Parkinson's. Consequently, the burden is really dramatic. So here you can see the results from a recent study from the Michael J. Fox Foundation, where you see the costs in Europe as well as in the US. And these costs are ever increasing with the ever increasing amount of elderly people and hence Parkinson's patients. So what we do at OrganoTherapeutics is using a different model system to develop new small molecules to treat PD. And particularly we use 3D cell cultures that mimic the human midbrain, so-called midbrain organoids. Here we can see some of those organoids in the dish, as well as the staining of a midbrain organoid. And we use those three-dimensional structures that we derive from Parkinson's disease patients to develop new small molecules. And I will go into more detail of some of those aspects in this presentation. So we see there are currently three main limitations. One that I already mentioned, that animal models as well as cell culture models are simply not predictive enough for the human brain. We address this problem by using patient-specific midbrain organoids, also called mini-brains, that recapitulate key features of Parkinson's. Furthermore, a lot of research and particular pharmaceutical research still is focusing on a relationship that there should be one compound addressing one particular protein or gene, which is a classical drug development concept. While we think that for complex diseases like Parkinson's, phenotypic assays make more sense. So we want to find a compound that stops dopaminergic neurons from dying. We are interested in the underlying molecular mechanism, but from our point of view, this is secondary. The main thing is the compound should stop those cells from degenerating. And the third limitation is that currently there's still a lack of stratification, though this whole idea that one compound should fit to all patients. While again, for a complex disorder like Parkinson's, we believe there needs to be a precision medicine approach, like we go for patient-specific or patient subgroup-specific models to precisely find the right compound for the right person. So our pipeline is to use Parkinson's disease patient-specific stem cells, which we get from a patient cohort here in Luxembourg. We differentiate those stem cells into our proprietary midbrain organoid model, where we identify disease-associated phenotypes and then test small molecule compounds to rescue those phenotypes. We use a lot of artificial intelligence approaches actually to really make sense of the enormous amount of data that we get with this approach. We use a lot of automation and high content imaging, which leads to really a lot of data, and only by applying machine learning based approaches we can really classify whether a particular organoid is more disease-like or is more healthy-like, and accordingly whether treatment with the compound can shift disease-like organoids into more like a healthy-like state. So that's how the midbrain organoids look like. These are three-dimensional structures consisting of a lot of neurons, particularly a lot of dopaminergic neurons, but also GABAergic and glutamatergic neurons, as well as a few serotonergic neurons. They contain support cells like astrocytes and oligodendrocytes, and in the second generation of these organoids, we can even enrich them with endothelial cells and microglia to mimic the immune system. We have published intensively on that. We have a granted patent which is licensed by the company. This is one of our key experiments. We took here stem cells from healthy individuals and Parkinson patients with a particular mutation and differentiated them either in 2D or in our midbrain organoids, and then simply counted the amount of dopaminergic neurons, while their loss is a key feature of the disease. In 2D, we cannot recapitulate this key feature. The patient has exactly the same amount of neurons as the healthy control. While in our midbrain organoids, in the patient, we see a very clear reduction in the amount of dopaminergic neurons. So the midbrain organoid is really recapitulating what's actually happening in the patient brain, and I will show you more examples of that. We can not only recapitulate the disease situation, which is the loss of dopaminergic neurons, as you see it also again in this quantification here, but we can also develop small molecule compounds to rescue it. So this is just an example study where we applied two compounds that were able to rescue the loss of dopaminergic neurons in those patients up to a level that's comparable to the healthy individual. And this study just recently has been accepted for publication in Science Translational Medicine and should come out within the next couple of weeks. Just to give you another example, where we again used patient-specific stem cells to derive midbrain organoids. And again, in the situation without treatment, the total amount of neurons is unaffected, but the amount of dopaminergic neurons is significantly reduced, as you can see here. However, upon treatment with a compound, we can rescue this loss of dopaminergic neurons to a good extent. And this particular compound, which is a variant of cyclodextrin, has been patented by us. However, our model is not only good for dopaminergic neurons, but it also captures other disease-associated defects, like defects in astrocytes. Here, we teamed up with a group of Nobutaka Hattori from Yotendo University in Tokyo, and they had post-mortem brain samples from Parkinson patients with a mutation in Parkinson. And what they saw in those post-mortem brain samples is that the amount of astrocytes is reduced here in the staining, as well as here also in the Western blot, and here quantified. Luckily, they not only had this post-mortem brain material, but also some stem cells, and we were able to differentiate these stem cells into midbrain organoids. And strikingly, again, we completely recapitulate what's happening in the patient brain. In the healthy individual, a lot of astrocytes are formed, as you can also see in the quantification, but in the patients with this Parkinson mutation, the amount of astrocytes seen in the staining and here in the quantification is really severely reduced. So an organoid of just a few weeks of age is recapitulating actually what's happening in a 50 or more year old patient. So we are very confident that this is indeed a very good model to mimic the disease, and then the next step to identify compounds that can rescue disease-associated features. We also developed our own compounds. So we conducted a first initial screen based on 640 natural compounds that we obtained from Griffith University. These are compounds isolated from plants and marine organisms, and we screened them for their ability to rescue autophagy defects. So we all know that in Parkinson's, autophagy is often strongly affected. So we were looking for compounds that could enhance the autophagy activity in Parkinson's disease patient-specific cells. And you see these individual dots here up this blue line are actually compounds that activate autophagy in patient-specific cells to a level that is as good as in healthy control cells or even better. From these 6 hits that we found in the screen, we were able to derive dose response curves, and in the next step, we would like to further develop these compounds in a classical hit-to-lead campaign. Let me just now go a bit about the development of our company actually. So we have been incorporated in July last year, and then moved into an incubator at the University of Luxembourg. In January this year, we obtained the first grant. We won a business plan competition in Germany and several other competitions, but probably the most prominent there is the Mars Challenge program that we're currently participating in. Since our incorporation, we were able to have a turnover of nearly 90,000 euro, and this is completely without any external investment. So this is really just from grants and ongoing projects. We have a good IP basis, so the main activities of the company are based on these 3 patents that we have licensed, but there is additional patents for compounds as well as for further development of the technology in the pipeline going to be filed within the next 12 to 16 months. There's an extensive list of publications about microbial organoids that we published within the last couple of years, and also there are more in the pipeline and going to come out relatively soon. And I mentioned particularly this Science Translational Medicine paper. There's several strategic partnerships that are important for us. We partner with the Luxembourgish Parkinson's patient cohort, where we get material. Then we have a partnership with the University of Luxembourg and the Luxembourg Center for Systems Biomedicine, embedding us in a strong Parkinson's research network, but also giving us access to state-of-the-art laboratories and platforms. And we have a very strong partnership with the Lead Discovery Center, where there's really experts in drug discovery and drug development, and also there's access to a chemical small molecule compound library. Our founding team consists of these 2 people. So on the one hand, there's Javier Giazzo, he has a PhD in biology and a very strong background in stem cell models, in vitro disease modeling, but also particular in the application of computational data analysis approaches like the machine learning-based image analysis that I've shown you before. And myself, I'm a professor at the University of Luxembourg, have a background in neuroscience, and particularly also in vitro modeling of Parkinson's disease. We have a strong advisory board consisting of persons with very diverse backgrounds, like we have, for example, Luis Almeida, who is a professor at the University of Coimbra, with a neuroscience background, Klebel, who is a CSO at the LDC, with a drug discovery background. Paul Lingor is a medical doctor with focus on Parkinson's disease. Manuel Gaviria is a serial entrepreneur in the neuroscience field, and Patricia Loquetta is a consultant in Luxembourg who was very strongly involved in establishing the Luxembourgish biomedical strategy. So coming to our business model, so in a nutshell, our main goal is to develop own small molecule compounds and out-license them. We see ourselves completely here in the preclinical stage, so we want to go from compound identification over optimization and preclinical animal studies, the preclinic. And then there is opportunities for out-licensing, where we would then generate income. This can be at the end of preclinic. However, if the situation, in particular the funding, allows, it could be also at later stages where we say we go 1 or 2 steps further to phase 1 or even phase 2 clinical trials. We have achieved already this first part here. I was talking very little about grants so far. So earlier this year, we were able to get a first grant from Johns Hopkins University about the further development of the Parkinson's disease model. And we have several grant applications with different programs in different agencies filed over the last 12 months, and we are very optimistic that one or the other of that is going to be granted within the next couple of months. We also conducted an analysis of the competitor landscape. There are several companies who use human brain organoid technology for different applications. Some companies like Axiosim and Stemonics are more focusing on service, while there is at least 2 other companies, System 1 in the US and AHET in Austria, who use brain organoids also for disease modeling. To our knowledge, they do not focusing on Parkinson's disease, but more on psychiatric diseases like autism and schizophrenia. However, they are definitely very relevant competitors because in principle, they could move into the direction of neurodegeneration, including Parkinson's disease. So we feel that for the moment, we have a strong position in the market, but we definitely need investment to grow and move quickly to secure our good position and further develop it. The good news is that there is indeed strong investment of venture capitalists into the Organoid technology. Like I just mentioned System 1, and they recently received a Series A funding of 25 million by this consortium here. So I think the community is really accepting and taking up the importance of the brain organoid system. So let's take a closer look at our financials. We are seeking an investment of 5.7 million, which would help us to bridge this part here in the preclinical study from the works that we have already achieved until the first point where we could house license. So this investment would really help us to conduct a hit-to-lead campaign with our existing hits, probably conduct further screening to get a broader basis of more hits, and then do further testing, toxicology, blood-brain barrier passing testing, as well as preclinical animal studies. Just to give you an outlook that our technology is not completely limited to Parkinson's disease. There is also other potential applications, and we started to explore a few of them. In principle, it's possible to use this model also on a service basis, like we could do research for others. We even could sell the midbrain organoids as products. We have a bit of work showing that midbrain organoids can be used for toxin testing, and this has been published. And this has applications in the civil area, like testing the toxicity of certain compounds, but also in the military area when it comes to testing of chemical weapons. Furthermore, our midbrain organoids contain not only the neurons that are relevant for Parkinson's disease, but also a particular neuronal cell type that's very relevant for schizophrenia. So also in vitro modeling of schizophrenia is something we could move into. And finally, there's a very interesting comorbidity between Parkinson's disease and diabetes. So we would be very interested in developing co-culture models of the brain and the pancreas to probably address both diseases into one system and also look at this comorbidity. So I think there's very exciting opportunities for further development. Just to sum up what I've shown you, OrganoTherapeutics is a spin-off from the University of Luxembourg. We use human patient-specific brain organoid technology with a focus on Parkinson's disease. We develop own compounds, and our business model is to out-license those compounds. We thereby address a clear customer pain point that there is a lot of failure in the pharmaceutical industry when it comes to development of compounds against Parkinson's disease, and our claim is that this is particularly the case because not predictive enough models have been used in the past. Our clear advantage is that our model really recapitulates key features of the Parkinson's pathology and that we have first proprietary drug candidates available. So thanks for your attention. Here are my contact details. I'm happy to answer any kind of requests and questions, and looking forward to hear from you. Thanks a lot.